Good evening. Thank you so much for joining us. I'm Andrea Piero, Director of the Visiting Artist Program at the School of the Art Institute of Chicago. It's my pleasure to welcome you to this evening's lecture by Suzanne Anker. Each academic year, SAIC's Visiting Artist Program hosts a variety of public presentations by internationally recognized artists, designers, and scholars with the mission to foster a greater understanding and appreciation of art and culture. Tonight's program is presented in partnership with SAIC's Conversations on Art and Science series. Under the leadership of President Emeritus Walter Massey, the Conversations on Art and Science series launched in 2011 as a forum for exploring interdisciplinary and critical perspectives on art, science, design, and technology. We are thrilled to have Suzanne Anker be a part of our rich history of distinguished guests who have spoken at the school, and I would like to thank her for taking the time to share her work with us this evening. At the end of the presentation, we'll have about 10 minutes or so to take a few questions from the audience before the program concludes by 7:30. Please raise your hand if you have a question, and our staff will circulate microphones for your use. We ask that if you're posing a question to please stand and share your name and to keep your question concise. It's my pleasure to now welcome to the podium Dr. Giovanni Aloi, Assistant Professor in SAIC's Department of Art History, Theory, and Criticism, to introduce Suzanne.
Thank you for coming. Simply put, if you know about bioart, you know about Suzanne Anker. I have the pleasure to work with Suzanne in a number of occasions. In 2015, we collaborated on an issue of my journal, and the issue was dedicated to Naturally Hypernatural, which was a conference that Suzanne organized at the School of Visual Arts in New York. It turned out it became 2 issues because there was so much great material we had to put out there. And I was very much touched when Suzanne turned up here at SAIC in 2017 to attend my botanical speculation symposium. We spent the whole day together, and the day before. It was very special to me also because Suzanne is a true pioneer of bioart, and there are so many interesting perspectives and ideas that she's been working on over time. So it felt like a very special privilege. And in 2018, I was invited to New York to the School of Visual Arts for the Hot House Archive Symposium that Suzanne organized on plants, because a lot of what brings us together and what has brought us together over time has to do with plants, which more recently, as you know, have gathered much more visibility in the arts. Before I tell you more about Suzanne and the amazing accomplishments that have characterized her career, I wanted to just give you a little bit of an introduction to bioart itself. Many of you who come here tonight surely have an interest in art and science, but bioart is a special thing, a specific weapon, I'd like to say. So, bioart can range from bacterial manipulation to glowing rabbits, petri dish sculptures, and the documentation of an ear prosthetic implanted on someone's arm. Bioart challenges disciplinary boundaries and institutional certainties. Its gestures and statements are always essentially political, in the sense that they never fail to change discourses, re-envision pasts, and question presence. Working with the living at the intersection of art and science, bioart is constantly busy asking important ontological questions, harnessing cutting-edge technologies, and at times inventing some more. Bioart knows multidisciplinarity like perhaps no other branch of artistic production. It deliberately disrespects territories and domains to subvert power relations, in the knowledge that the relentless blurring of disciplinary boundaries can yield truer knowledge than the kind produced by compartmentalized subjects. Its conceptual strength lies in the acknowledgement that art can be particularly useful, that it should be impermanent, and that it must be uncontainable. And bioart loves, I suspect, to give museums an almighty headache when it comes to archiving, maintaining, and staging works of art. It is this irreverent slant and provocative attitude that enables bioart to push the boundaries of cultural discourses at a time in which, more than ever before, we absolutely must rethink our boundaries, our territories, our ability to collaborate across disciplines, and with that, our futures. So, as I said earlier, Suzanne Anker is a true bioart pioneer, a visual artist and theorist working at the intersection of art and biological sciences. Her practice investigates the way in which nature is being altered in the 21st century. Concerned with genetics, climate change, and extinction, she calls attention to the beauty of life and the necessity for enlightened thinking about nature's tangled band. Anker frequently works with predefined and found materials, botanical specimen, medical museum artifacts, laboratory apparatuses, microscopic images, and geological fragments. She works in a variety of mediums ranging from digital sculpture and installation to large-scale photography, to plants grown by LED lights. Her work has been shown both nationally and internationally in museums and galleries. Anker's exhibitions have been the subject of reviews and articles in the New York Times, Artforum, Art in America, Flesh Art, and Nature. Her books include The Molecular Gaze, Art in the Genetic Age, co-authored with the late sociologist Dorothy Nelkin. She has hosted 20 episodes of the BioBlurb Show, a program originally on WPS1, Art Radio, in collaboration with MoMA. Chairing New York's School of Visual Arts Fine Arts Department since 2005, Anker continues to interweave traditional and experimental media into her department's new digital initiative and the SVA BioArt Lab, where students work with the tools of science to make art. Suzanne Anker, everybody. Thank you very much.
Thank you for having me. So we're going to speak about a number of things today that seem to resonate with the current time. And time is one of those concepts in which we look at as before time, before this time, and after this time. And currently, we are experiencing a rapid epidemic or pandemic, which is very reminiscent in many ways of the AIDS epidemic that we have experienced, because AIDS represented a period of time before sex without protection and then after AIDS sex with protection. So it raises this question about a virus in a globalized world and drives home the point that the world has no separation. And this is something that we need to consider from a political, social, economic, artistic, and cultural perspective. So today's lecture will kind of look at some of my work, which aims at investigating the afterlife of particles, which is a very different notion of discrete entities as Aristotle had speculated. We now can alter nature in numbers of ways, some for the good, some for the bad. And we are now waking up to the fact that we have to steward nature. So let's take a look at a couple of ideas here.
A new generation of biohackers is tinkering with DNA to create plants with new colors. At the School of Visual Arts in Manhattan, plant researcher Sebastian Cachobo is trying to create genetically modified plants, mostly from information he learned on the internet. One of his pursuits in the school's bioart lab? the blue rose, considered the holy grail of horticulture.
I think the main reason why people are trying to make blue plants is because there are very few, if not any, blue plants out there that are manipulatable or growable in a specific condition. Basically because those high saturation colors just simply do not exist in nature.
GMOs, or genetically modified organisms, have drawn a considerable amount of controversy, with concerns about the potential impact on health and the environment. Cachobo doesn't think such worries apply to projects like his.
The main argument I would have towards anybody who would contest us going through with this project is the fact that it'll be contained, and it's a plant. So we're not working with pathogens or any other type of human or mammalian tissue.
Suzanne Anker sees the process as more of an art project than a science experiment.
Artists here use science as a tool, so they are not engaged in research in the same sense that a scientist would be. Blue is kind of an area of wonder. Children say, why is the sky blue? All the time. That's one of their first questions. And I think that is in part the background of this. But I do think that even if it doesn't work, it doesn't matter because it's art.
The process is meticulous and complex. It all comes down to identifying and manipulating a specific gene that could give the plant its color.
The gene in question, called AE blue, produces a beautiful cerulean hue in bacteria. It's never been tested in plants as far as I know. So this will be the first time that we actually try to put it into a plant.
With the project about halfway done, Cachobo is still tackling problems, but he says even if it fails, the experiment won't be a loss.
In an ideal situation, in another 3 months, we'll have our blue plant. Worst case, it might not work at all. And that's the risk that we're willing to take, and that's the risk that we want to attempt, because even a failure might be useful in terms of understanding the processes at hand.
This piece was on the front page of the Wall Street Journal, as well as this video clip on their site. Not very often does an art school get to be featured in that way. But what is really important here is the idea that if it doesn't work, it doesn't matter because it's art. What did I mean by that? Well, I mean that for every scientific innovation, there's a parallel cultural component. So if we look at the blue rose and we look at that idea in the historical literature, we come back to Novalis, the philosopher and poet, who is talking about the blue rose as a symbol of the infinite, of the unknown, etc. And the blue rose has appeared and reappeared in the writings of Walter Benjamin, in the films of David Lynch, in Philip K. Dick's work, and as well as Stanley Kubrick, in which he uses this blue flower in Eyes Wide Shut to have one of his protagonists, while dancing with Nicole Kidman, show off this blue flower. So again, the appearance and reappearance from philosophy to literature and to art is what we're talking about, and the fact that if it doesn't work, it doesn't matter, because it will be part of the cultural imaginary. And if we look to the scientific establishment with regard to the blue rose, we get to see in 2009 a company called Florigene named the blue rose Applause. And it was genetically modified to produce delphinium, which is a pigment found in most blue flowers. Well, to everyone's surprise, the blue rose was not blue. It was lavender. So this is still a major kind of unknown within botanical circles, as well as philosophical, literary, and artistic circles. One of my pieces from 2015 was Astroculture, Eternal Return, which was shown at the Cathedral of St. John the Divine in New York City. Here you can see, if you shine a light onto the vegetation, you see that the plants are really green, making fuchsia the new green. What the red and blue lights do that emanate this color is to create that part of the light spectrum that is responsible for photosynthesis. So we are able to kind of tap in to a kind of photogenesis in order to grow our plants indoors. And I traveled around with this exhibition in a number of places, as well as the Daejeon Biennial in Korea several years ago, and the museum had turned the entire biennial over to bioart. And these are some of the images of the plants sprouting there. And what I had to do in Korea is essentially buy Korean seeds and Korean soil because you cannot transport living matter from one country to another without essentially a plant passport. So this was very interesting to me to work in Asia to create this piece. Now, the idea comes from NASA, who had put astronauts in space. The first time they did that was in 2009, in which Japanese astronauts went up to grow cucumbers. And they found that everything we know about botany on Earth is different in anti-gravitational conditions. So on Earth, if you plant a seed, you get one sprout. In space, if you plant a seed, you get two sprouts. If you boil water in space, you get one giant bubble. And if you smell a rose in space, it smells differently. So this is part of what our future will be like as we explore other planetary possibilities. So here you see the astronauts who are eating their space lettuce, and the 2009 astronauts who grew cucumbers were not allowed to eat those cucumbers because it wasn't clear whether they would have certain kinds of microbial infections. This is the exhibition in Daejeon, and you can see that everything is working perfectly well, which I was happy to say, and that the sprouting conditions actually worked well. Okay, and another piece of mine, which is also an ongoing piece, which refers very much to the destruction of coral reefs worldwide. This piece is called Biota, and these are handmade porcelain sculptures that use a natural sea sponge as its matrix. It is then dipped into liquid porcelain and burnt out. The sponge burns out, leaving the residue of itself as a kind of ghost phenomenon, and represents here not sponges so much as dead coral. Now, the sponge is a really interesting animal because it is the first multicellular animal that we can look towards and is now being used as a model organism for neurological disorders. The sponge has all the equipment to make a nervous system, but never did. So the question about what we can learn from that for treating diseases such as Parkinson's and ALS is something under investigation, because we share 70% of our genetic material with the sea sponge. Now, we don't look like the sponge, it doesn't look like us, but we are very close relatives. And here you can see a close-up of the kinds of detail that one can get using this very classical material in which every kind of polyp in the sponge can be representative. The sponge, also being a multicellular animal, shows you how a community, essentially, of parts can work together. Each one of these sponges is unique, except for this one, which is actually a coral. And I thought making the comparison between the real and the simulated would be a good idea. I also used some of these little figurines to talk about the way in which consciousness erupted with the move towards figurative art from the Neolithic to the Paleolithic time frame. Because there have been also theories about the fact that the brain changed when man became upright, and what essentially happened was figurative sculpture was produced. Now, the sponge is a great metaphor for the brain as well. Even Emily Dickinson talked about that in many of her poems. This is a recent show that I did at the Everson Museum called 1.5 degrees Celsius, which is a reference, of course, to global warming. And on one part of the installation, one sees the biota pieces on plexiglass, and the other part of the exhibition, there are many photographs, and they're quite large. They're 44 by 44 inches that are placed on the wall. And these photographs are representations of still lifes that I constructed in Petri dishes that combine something from the natural world and the artificial world. So as a reference to synthetic biology, what we see here are combinations of different kinds of materials that really are being explored in the laboratory, but these become a kind of poetic ode to the recombination of matter, which is very much what we're doing now. Synthetic biology is also a reworking of the word genetic engineering. And I don't know how many of you are familiar with CRISPR, which is a gene editing program now that is being used by scientists and by the general public. And it's sort of questionable in the sense that it assumes that one gene and is responsible for one effect, when in reality our genes act in concert. There's an interesting program on Netflix for those of you who want to learn more about this and get scared about this. It's called Unnatural Selection, and it is about Josiah Zahner, who has started a business that you can order one of these CRISPR kits and do CRISPR yourself. It's extremely controversial and very entertaining and extremely scary. You know, what was computer hacking in the 1990s is now biohacking. And there are many do-it-yourself laboratories throughout the world in which you can conduct your own kind of experiments. You don't have to be connected to institutions. And the idea was to democratize science. But like all things in a democracy, you get a kind of a curve that moves from those who are who take their job seriously to those on the fringe. So this is now being very questioned by many authorities. Vanitas in a Petri dish. Okay, what is vanitas? Vanitas comes from the art historical genre in the 18th and 19th century, in which people in Holland were considering what materialism did to their spiritual involvement, and painted a number of paintings that had things that expired, such as wilted flowers, or skeletons, or musical instruments that no one was playing. So Vanitas in a Petri dish is an update on that idea, kind of questioning what the ethics are in biotechnology, as we can now eliminate discrete species. We can put firefly genes into tobacco plants. We can put flounder genes in tomatoes. We can mate animals that normally could not create an offspring. So what does this mean to alter nature in terms of our ethical responsibility? And this set of vanitas, and here you can see an egg and a little crab, and it almost looks like a planetary composition. Here one sees metal and mushroom and plastic recombining in a form of a three-dimensional collage to essentially reference the way we can cut and paste information that is living, so to speak, and create new entities. I think in these pieces it's very clear that we live in a second nature as well as our primary nature, and that in our second nature we use the colors of our surroundings to create artificial entities. So here we have a sunflower and a rubber ducky, practically the same color. And in the piece on the right, we see flora coupled with a geological item up here, as to think about what we can think about the relationship of flowers and crystals. What would happen if flowers could spawn crystals? These are more about the perception of visual imagery. So the piece on the left is some meat and some dead flowers and some little baby rocks, as well as these images up here, which are cut out flowers. So these are not real. They're put together with other aspects of the natural world. And one can kind of ask the question is, what is our relation to simulation? What is our relation to the synthetic? What is our relation to the artificial? And the artificial keeps on cropping up. There are now artificial eggs, for example, some of which are very tasty, as according to one of the Bill Gates, that's made from a bunch of stuff and tastes like eggs. But then there's artificial eggs that are made from glue and non-nutrition information that is being sold in markets because they're cheaper. So we've seen the Bear Burger and the Impossible Burger, and all these burgers have the same calorie count as a real burger, but they are operating on a different kind of notion in the sense that there are environmental concerns there. And the piece on the right, which is a shrimp and a metal item and some broken glass, again combines the inorganic with the organic. So vanitas in a Petri dish goes back to art history and brings it up to the 21st century. But what I thought I would do after I took all these photographs was run them through a 3D program to see what would happen if I created a z-axis. So I took the pea pods and the egg and the mushrooms and flowers, created a three-dimensional axis, and what did I get? I got a micro landscape. Now, I had no idea that this was going to happen, but after it did happen, I continued with this project to kind of see what else could be articulated. And one of the things that really surprised me here were some of the edges, you can see it a little bit here, in the sculptures, in which the machine learning of the program had to make certain assumptions. And these are assumptions that I never made. So I learned from the machine learning myself about what other possibilities there were for edges in sculpture. And here you can see the vanitas in a Petri dish, as well as the remote sensing pieces in this, what's called a Frank case. And there's a one-to-one relationship here between the little sculptures and the large photographs. So when visitors come to the museum, they try to match up. This relationship becomes sort of a game. Now, the name remote sensing comes again from NASA, in which it is about satellite data gathered to look at spaces that are either too war-torn to visit personally or too toxic or for surveillance, etc. So it is a topographical view. And we've seen that operate in art history as well, starting with people going up in balloons. And so this question of changes in vision based on instrumentation has always been part of my work. And in this one, you can see that the glass vessels also act as devices to change what is being seen, as if they are lenses. And we saw the egg and we saw the rubber ducky two-dimensionally, but three-dimensionally, it's a very different circumstance. And here you can see the edge, as I was talking about. Look at this up and down edge that the machine taught me. And this one is on our poster, and it is a micro landscape based on a cookie and some flowers and some coral. And again, the zinnia is now almost an amphitheater. So this question about metaphors and how we look at metaphors, not only in what is redeemable about them, but what they tell us about the culture at large. And I think that currently, again, going back to the virus, is that throughout history, the virus has always brought with it metaphors of disease. So in the 19th century, we have the metaphor of tuberculosis being a disease that infected sensitive writers and artists. In the 20th century, we have AIDS, which was considered a disease based on immoral behavior. We have also cancer, which has been considered a disease for those people who could never express their emotions. So we're struck with this again now. What is the coronavirus going to be, the metaphors for the coronavirus? And the coronavirus is really a virus of globalization and what that means. We've never really known what globalization is, and now we can see a little more about that. But let's go back to some sculpture. Okay, this is called Carbon Collision of the Diamond Mine. This again is porcelain, and it is made from tube sponges. And every detail in the sponge, every little prickly extension is able to be produced in this transformation of material. And it's important to think about transformation in the broadest sense, the way in which a microbe can transform a society, the way a material can transform a metaphor, the way in which a genetic sequence can transform a disease. And here are some photographs of what appear to be underwater photography. Well, they're not. okay? These were produced in a Petri dish from some goop that was taken from the underside of a pier at Woods Hole in Massachusetts. And what we see here now are some tunicates, which are single-celled organisms, and we appear to see the under-the-sea notion as something that is galactic. So the issue, too, of transforming the micro to the macro is one way to produce knowledge. This is the BioArt Lab. It was founded in 2011, and every year we have a residency program, and people from all over the world, undergrads, grads, people who are out of school, come and visit us to work on projects such as working with biomaterials, mushrooms, working with SCOBY, which is a byproduct of kombucha that produces material that looks like latex or leather. We also do tissue engineering of plants. We also work with some ecopoxy. And what nevertheless happens, and this happens in my bioart class as well, is the discussion always turns towards the philosophical implications of altering nature. Okay, this doesn't happen in a painting class. It doesn't happen in a drawing class, but it always happens in a bioart class. And here you can see we have a lab that is a cross between a 19th century and 21st century lab. We have plants, we have animals, we have a molecular corner where we do DNA testing. We've done DNA testing of foods to find out if they were GMOs. And we invite scientists to come in to work with our students, and we invite art historians, and we invite people from literature and philosophy. And what's interesting when the scientists come in, since it's not a typical scientific lab, they're very tentative in approaching what they're doing, because part of the impetus of creating this lab was to allow artists the freedom to work with scientific tools. As many scientists are very skeptical about artists coming into their laboratory, because artists notoriously break all the rules. So this is an opportunity. It's a space where one can freely develop projects that scientists would never think of. And here is one project, for example, that we did. All literature said that you can't grow baby skates or hatch baby skates in captivity. Well, we did it about 8 times, and this has become our little mascot. And what's interesting about the baby skate is transparent when it's very, very young, and you can see all of the organs inside. But really our tour de force was a project we did for the Biodesign Challenge. We entered 2 competitions. One was iGEM, which is a synthetic biology competition that's divided into categories based on material or code, etc. But we were in the art and design category for our project, and we won a gold medal. An art school won a gold medal. And our project there was called Soiled. We went around New York City and did soil sampling for what kind of nutrients were in people's backyards, and we created a device that would allow us to use less toxic indicators. So soil sampling is based very much like on color-aid paper. There's a parallel there in which you use color as an indicator relating to an element, whether it's magnesium or aluminum or manganese, etc. So that was our first competition. Our next competition was the Mayo tomato, which has been shown in many, many places, including MIT. And the concept here, and this is from 2016, this is before the Impossible Burger. We thought if we could genetically engineer a tomato to carry protein, then you could just plant your seeds. You wouldn't have to kill any cows. You wouldn't have to essentially increase methane in the environment, and you would be able to get the nutritional value that you want. So myoglobin is a protein like hemoglobin, okay? And it is, this whole idea stemmed from the pun of the beefsteak tomato. And so this is our beefsteak potato called the Mayo tomato. And I'll go over some of the other images. We also created a whole series of product line and recipes of theoretically what you could do with this Mayo tomato. So this is a projected growth of demand of beef. I think it's from the United Nations, but what's really weird is India. They don't even eat beef. So I don't know what this map really is about, but you can see that the growth of the meat industry is upon us. So the cow part's anatomy, goodbye. And here we have a meatball as a representative of something that we don't need anymore if we have this tomato. Okay, we had some scientific help, and this is how we did this. We essentially did some protein research. We also synthesized DNA and assembled it. We exposed the plant tissue to bacteria that we used as a vector. We tissue cultured the little plant, and we grew it so it had baby plants. And we analyzed for total protein content versus the natural tomato. So what we did find was that we did get the myoglobin gene into the tomato, but we didn't know how much protein, and we didn't know where it was in the tomato. So essentially, pretty much that's as far as we went. If anyone in my class wanted to get a PhD in botany, they would continue it on. But this was a speculative project, and as such, it made a tremendous amount of sense. We also made Mayo tomato jerky, Mayo tomato soup as a kind of reference to Andy Warhol. This was our installation here that we had photographs, sculptures, scientific kinds of gel charts, etc. And then what we did has created this video.
Hi there. Welcome to the SBA bioart lab, where we've been designing the future of food for over 30 years. Good. This laboratory, the first of its kind, is where biologists and artists work together tackling some of the world's biggest biological concerns. Over the years, we've spawned some unique results, but nothing as successful, impactful, and delicious as this. Meet the Mayo tomato, the amazing protein-packed fruit that started it all. A biological breakthrough that changed the way we think about protein. I have something amazing to show you. Randy, get the thing.
As you wish.
Before the Mayo tomato, humans relied heavily on animals for their daily source of protein. With a growing global population and a strained agricultural system, we recognized animal protein was not an ecologically sustainable source. There had to be an alternative method. We put together some of the brightest scientific and artistic minds we could find, and the result was impressive. Take a look.
This will only hurt a little bit, JK.
In 2016, the bioart team at the School of Visual Arts engineered the world's first fruit to produce the same protein, myoglobin, found in animals, the Mayo tomato. They found plants to be deficient in megadoses of protein necessary for a nutritional human diet. So they translated a DNA sequence found in beef and inserted it into a tomato's genome. The result? One little tomato possessed the same amount of protein found in an 8-ounce steak. This breakthrough creation caused quite the stir. It seems not everyone was a fan of this amazing scientific achievement.
GMOs are dumb and stupid and poison.
Of course we're not happy.
However, in the year 2030, mad cow disease almost completely wiped out the cattle industry, creating an urgent need for the Mayo tomato's amazing protein power. And soon, hearts, minds, and mouths were open. The Mayo tomato ushered in a global movement that ultimately ended our need for animal consumption. Not bad for a group of art students, if you ask me.
Great. So these are sort of some of the ideas inherent in bioart, which is in itself a contested term. Many people have taken credit for coining the phrase, but no one really knows where it came from. There's also no clear definition of what the parameters are. Some people would take the opinion that it has to include live material. Well, that would mean that someone who has a cold and is sitting in a gallery is doing a bioart project. So that definition, I think, needs to be expanded. And when I wrote the definition for the Oxford Dictionary of Art online, I divided bioart into 3 subgenres. One which had to do with a molecular view of matter. And that molecular view pretty much was about instrumentalized vision, and one could use any medium whatsoever, whether it's film or painting or drawing, etc., because it was about tapping into, again, the cultural imaginary and dealing with the philosophical implications of altering nature. A second subgenre would be computer-generated art, because biology is technology, technology is biology. And without technology, we would never have been able to decode the human genome and what followed. So that instrumentation, again, is part of the result. Freeman Dyson died this week, who was an august physicist, and he always talked about the fact of scientists inventing new tools in order to create new discoveries. And so digitalization is critical here in bioart, to 3D model things and to process biological information in an orderly way. And the third aspect, of course, is wetware. And wetware are the experiments that happen in laboratories, perhaps using live material, whether it is bacteria or mushrooms or plant matter, etc. So I think that opens the field to a much larger discourse and resonates much more with the history of art, because art has always been involved with aspects of nature during different periods. So one could look at bioart as a really late phase of Romanticism, in which the subjectivity and idea of the individual is kind of the driving force behind innovation. I think speculation is a great tool to uncover new practices and should be considered a form of research itself. So thank you very much. Do we have some questions?
Hi, I'm Andy Scarpelli. I'm the lecturer for bioart here at the SAIC. I'm also the founder of Chi-Town Bio, the DIY bio movement here in Chicago. And so I had a bunch of questions about this. This was a great lecture, and I really loved hearing your views on bioart. One of the things that really surprised me was you were talking about your kind of suspicion, maybe, or dubiousness towards the idea of the DIY bio movement and the ethics involved with it. And I think that fascinates me. I came as a biologist into bioart, kind of from the opposite angle that I think a lot of people come towards bioart. And I think bioart and what you said earlier about the philosophical coming into bioart is really potent. And so my question really comes down to, I want to really hear your views on how bioethics colors how bioartists view their material and their work, simply because I think the DIY bio movement has a lot of different viewpoints on bioethics, and there's a lot of that in bioart too, when you go from Zaretsky to a bunch of other artists that are really pushing that edge. How does bioethics especially play into your view of what you tell people about bioart?
And this is a very good question, and I'm going to sort of try to answer it with a question. Is there such a thing as fraud in bioart? Many bioartists have assume to tell us that X, Y, and Z is true when, in fact, there's been no peer review nor documentation about whether or not this experiment is true. I just came back from from North Carolina, where there was an exhibition there about our genetic futures. And what was really interesting there was the discussion I had with several of the scientists who were working also with CRISPR, because that's where it comes out from, University of North Carolina in Raleigh, as well as gene drives, which are constructed to essentially reduce populations of what are considered vermin, whether they're mosquitoes or rats, etc. And in this conversation, they came up with a project that they were working on to codify laboratory practices in the do-it-yourself bio community. This will be a free online document of protocols that will not necessarily enforce procedures, because the DIY community is essentially independent, but to essentially establish some rules about bioethics. When I mentioned Josiah Zahner, he was in California with his crowd of people, in which he injected himself with CRISPR to essentially grow better muscles, okay? And the people in North Carolina, scientists as well as Daniel Grushkin, who is from GenSpace in New York, were horrified by this. So the question of, bioethics is one about do no harm. And I think that one needs to be responsible for the messages that they're sending out. Now, that gets into this whole idea of censorship and what that means, but I think that there needs to be more discussion about what is going on in this way, because there have been many really great bioart projects. Another great project was a woman in Amsterdam who tried to make bulletproof skin, and she worked with silkworms and scientists, etc., and it almost worked. Now, this would never be a project that a scientist would do, and there are many others like that. So the need for this kind of speculative innovation is really critical, but I don't think it should become dangerous to an uninformed public. Does that answer your question? I mean, you can't rely on artists to be bioethicists, okay? Bringing a group of artists together to agree to anything is like herding cats. So I think you need, again, this multidisciplinary kind of platform where you bring people in who are experts in these different areas. There's a...
Hello. I wanted to ask you, since you spoke a bit about sea sponges and their similarity in like their genomes to humans.
Could you repeat that? I can't hear you.
Oh, sorry. Can you hear me now?
Yeah.
Okay. I wanted to ask you a bit about sea sponges since you brought them up, and their similarity in their DNA type to humans. And basically their significance as organisms. So I'm curious how you personally feel about using them and kind of like sacrificing them for your work that is discussing the destruction of another living organism.
Okay, this is a very good question. These sea sponges are used. They come from eBay, so they will be discarded anyway. So in a certain sense, I'm reclaiming them for public vision to help people think about the destruction of coral reefs, etc.
Hi. I actually have like a two-part question. I was wondering what some of the challenges of starting a biolab are in terms of funding and how you organize that type of environment. And my other question was how you kind of work in a space that has a lot of hype and how you kind of avoid some of that hype with your work, just trying to get to the core of what's important and kind of cut out some of the things that are making a lot of headlines, but maybe not as important. I don't know if that makes sense, how I phrased it.
Okay, well, first of all, our lab is a level 1 laboratory, which means we don't deal with anything infectious or HIV or body fluids, etc. So we have a very strict protocol in the lab. The lab was started in 2011 as an extension of all of the new technologies that I brought to the department. I initially started with things like rapid prototype, laser cutting, video mapping, digital sewing, etc. And it made sense that a biolab would be an extension of that. School of Visual Arts is a private institution, so the way we are funded as a for-profit school is based on how many students are in my department. So I get a certain amount of money for each student in my department that I can use in ways I see fit. So there's no fundraising here, which is why I was able to do this. And when I first went to the president of the school and said I wanted to construct a bioart lab, he didn't know what I was talking about, but he let me do it anyway. And now when prospective students come, particularly their parents, and they see the bioart lab, they are very pleased that there is some sort of academic aspect to fine arts. As far as what we do in there, and it changes every year, is we try to conceptually address what the pressing issues are with regard to the intersection of biology and citizen science. So that fact that science has spilled out of the laboratory into our lives, from the clothes we wear, the food we eat, diseases we get, the medicines we take, etc., that it seems as if in a time of global crisis and species extinction and climate change, etc., that we need to step up to the plate. So that's really our mantra.
Thank you, Suzanne. My name is Ame Kataria, and my question is about the Mayo Tomato project. Why did you choose a tomato to transfer the myoglobin?
Why did we choose a tomato? Essentially, we chose a tomato based on the fact that there is a variety of tomato called beefsteak. So that's where the idea came from as a pun on beefsteak. Also, the tomato has a really interesting origin. It kind of starts in South and Central America, goes to Italy, goes to Europe, and then comes back to the United States. It didn't come from South America to North America. And the theories of the tomato was that it was poisonous. And there's a lot of anecdotal kinds of discourse around the tomato, because all parts of the tomato plant, except for the tomato itself, are poisonous. So the roots, the leaves, the stems can all kill you if you eat them. And there was this kind of anecdote about someone who tried to kill George Washington by putting tomatoes in a salad, and he didn't die, so Americans adopted the tomato. I mean, it could be any vegetable. I mean, another idea would be duck l'orange, for example, where you take a gene from a duck and put it into an orange. Or fish and chips, where you take a gene from a fish and put it into a potato. So there are many kind of speculative ideas, but because the beefsteak tomato actually is named such, that's where we got the idea. Okay, thank you.